So shall we start, sir?
Yes, sir.
ಪ್ರಾರಂಭ ಮಾಡೋಣ ಸರ್. ಓಕೆ ಸರ್. ಎಲ್ಲರಿಗೂ ಶುಭ ಸಂಜೆ. ಗುಡ್ ಈವ್ನಿಂಗ್. ಇವತ್ತು ಪ್ರತಿಭಾಶೋಧ ಉಪನ್ಯಾಸ ಮಾಲೆಯಲ್ಲಿ ನಮ್ಮೊಂದಿಗೆ ಡಾಕ್ಟರ್ ಅರುಣ್ ಇಸ್ಲೂರ್ ಸರ್ ಇದ್ದಾರೆ ನಮ್ಮ ಜೊತೆಗೆ. ಈ ಉಪನ್ಯಾಸಕ್ಕೆ ಅವರು ಬಿಡುವು ಮಾಡಿಕೊಂಡು ನಮ್ಮೊಂದಿಗೆ ಸೇರಿರೋದಕ್ಕೆ ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿ, ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಇಲಾಖೆ, ಕರ್ನಾಟಕ ಸರ್ಕಾರ ಹಾಗೂ ನಮ್ಮೊಂದಿಗೆ ಇವತ್ತು ಸೇರಿರುವ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳು ಹಾಗೂ ಶಿಕ್ಷಕ ವೃಂದದವರಿಗೆ ಸುಸ್ವಾಗತವನ್ನ ಕೋರುತ್ತಾ ಇವತ್ತಿನ ಒಂದು ಉಪನ್ಯಾಸ ಮಾಲೆಯನ್ನು ಆರಂಭಿಸೋಣ. ಈ ತಾಂತ್ರಿಕ ಉಪನ್ಯಾಸವನ್ನು ಆರಂಭಿಸುವುದಕ್ಕೆ ಮುಂಚೆ ಡಾಕ್ಟರ್ ಇಸ್ಲೂರ್ ಸರ್ ಅವರ ಬಗ್ಗೆ ಒಂದು ಕಿರು ಪರಿಚಯವನ್ನು ಮಾಡಿಕೊಡಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ಡಾಕ್ಟರ್ ಇಸ್ಲೂರ್ ಸರ್ ಅವರು ಬ್ರುಸೆಲ್ಲೋಸಿಸ್ಗೆ ಎಲಿಸಾ ಪ್ರಮುಖ ಮಾಸ್ಟೈಟಿಸ್ಗೆ ಕಾರಣವಾಗುವ ರೋಗಕಾರಕಗಳಿಗೆ ಮಲ್ಟಿಪ್ಲೆಕ್ಸ್ PCR.
Sir, excuse me, sir. Actually, brucellosis ನಮ್ಮ elder brother sir, Dr. Shri Krishna Isloor.
ಹೌದಾ ಸರ್? ಓ ಸಾರಿ ಸರ್. ನಮ್ಗೆ ಸಿವಿ ಇದು ಸಿಕ್ತು. ಜಸ್ಟ್ ಅ ಮಿನಿಟ್ ಸರ್.
ಓಕೆ. ನಾನು ಕೆಮಿಸ್ಟ್ರಿ ಅಣ್ಣ ಬ್ರುಸೆಲ್ಲೋಸಿಸ್.
ಜಸ್ಟ್ ಅ ಮಿನಿಟ್ ಸರ್, ಜಸ್ಟ್ ಅ ಸೆಕೆಂಡ್.
ಆಯ್ತು ಸರ್, ಆಯ್ತು. ಇದು ಒಂದು ಕೆಲಸ.
Sorry for the inconvenience caused.
No problem, sir. No problem.
Sir has completed his BSc in Karnataka University in the year of 1996, MSc in the year of 1998, and PhD from Mangalore University in the year of 2002. Sir is currently working in the National Institute of Technology, Karnataka, since 13 years. And his areas of interest are membrane technology, medicinal chemistry, nanomaterials, organic synthesis, organic electronics, and his significant projects are membrane technology and organic electronics. He has supervised more than 13 students in PhD under his guidance, and he has published many reputed journals in national and international journal papers. His achievements are: he is the best reviewer in European Journal and Medicinal Chemistry for the year 2013. The best reviewer award has received even in desalination for the year of 2012, and he has received a best research paper award from VGST, Government of Karnataka, during 2012, and BRNS Young Scientist Award during 2009. Apart from that, he has published more than 100 research papers in ACTA, Crystallographica, in International Journal, and received prestigious Technion postdoctoral fellowship from the Technion Israel Institute of Technology during 2005 to 2007. Apart from that, Sir is Managing Secretary of NITK in English Medium School from 2013 till date, and served as one of the active warden of NITK hostels and faculty advisor of IST students chapter at NITK and faculty advisor of Management Forum of NITK from 2012 till date. With this brief introduction, I welcome you once again, sir. Over to you, sir.
Thank you, sir. Thank you very much for this opportunity. I hope I am audible to all of you.
Yes, sir.
Okay. Can I shift to the slide mode now? Sir, I hope the slides are visible.
Yes, sir. Perfect, sir.
Yeah.
Go ahead.
Okay. A very good afternoon, one and all. And first of all, I would like to thank the KSTA team for giving me this opportunity to present this organic molecules before our under the talent search program of the KSTA. And first of all, I would like to give my acknowledgements, sincere acknowledgements to Dr. Ramesh and his entire team of KSTA, then Director of NITK Suratkal, and then various funding agencies such as Department of Atomic Energy, Board for Research in Nuclear Sciences, Government of India, DRDO, Government of India, Department of Information Technology, Government of India, Vision Group on Science and Technology, Government of Karnataka, and Ministry of Mines from Government of India for their generous funding. Probably without their generous funding, we would not have achieved this much in our present research. And of course, I would like to thank all my former as well as present group members. Already about 18 candidates, they have successfully passed out from my group with the PhD degree. And currently about 7 more are working. It's because of their hard working as well as their uh, contribution. The whatever number of publications or patents are there, I would like to acknowledge my group members also on this occasion. Yeah, without making any further delay, let us focus on to the world of carbon and the molecular diversity of the life. Dear students, you may be aware the carbon is the backbone of the life. You take any molecule in our universe, either it may be a amino acid or it may be a protein or it may be any of the nucleic acid or it may be the carbohydrates. You name any of the or maybe the lipids, any biomolecules. You take any biomolecules which is composed of carbon as a one of its backbone, or at least the carbon atoms which are present. So all the living organisms consist mostly of carbon based compounds, and carbon is unparalleled in its ability to form the large complex and diverse molecules. As I mentioned, the proteins or the DNA or the carbohydrates and other molecules that distinguish the living matter all are composed of the carbon compounds only. And the carbon is important enough to have its own branch of chemistry called as organic chemistry. As you know, the carbon organic chemistry, it mainly deals with the study of carbon molecules only. However, this particular talk, it is actually highlighting about various type of carbon molecules, carbon-based molecules as part of the organic chemistry. And these organic compounds, which range from simple molecules to the very complex one. Say the simple molecules, like it may be like a methane, which is the one of the simplest hydrocarbon, and it may be highly complex molecules such as varieties of olefinic molecules or other higher hydrocarbons. So it such a large diversity we can find in the carbon compounds. And most of the organic compounds, they contain hydrogen atoms in addition to the carbon atoms. Along with, there may be presence of other heteroatoms, like it may be the oxygen or nitrogen or sulfur or phosphorus. And whenever a cyclic compound which contains other than hydrogen, carbon and hydrogen, when there is presence of oxygen, nitrogen and other heteroatoms, they are typically called as heterocyclic compounds. And about more than 90% of the drug molecules which are commercially available, they are composed of heterocycle. That means again, they are also basically carbon only. And of course, different theories have been proposed to uh, highlight how this carbon was originated on this planet. Among the different studies, I would like to mention the most accepted theory, that is nothing but the Miller Urey experiment, which was demonstrated the abiotic synthesis of the organic compound. It's a simple experiment. I'll just take 1 minute to explain about this particular experimentation, which was proposed by Miller Urey. In a glass container, uh, some calculated amount of water, and then methane gas, and then ammonia, and then hydrogen. These were sealed, and then connected to the glass tubes, and the flasks were connected to a loop with one flask half full of liquid water and another flask containing pair of the electrodes. So this is the typical diagram which you can see the experiment. And the liquid water was heated to induce the evaporation, and also sparks were fired between the electrodes to simulate lightning through the atmosphere and water vapor. and then the atmosphere was cooled again so that the water could condense and trickle back into the first flask in a continuous cycle. That is nothing but it is just like an artificial lightning type of the arrangement which was made through this particular experiment. And within a day, this particular experiment was continued for about almost 18 to 20 hours. And within that particular 18 to 20 hours, the mixture has turned into a sort of a pink color. And at the end of 2 weeks of continuous operation, further this was continued again for 2 more weeks continuously. After about 2 weeks, Miller and the Urey, they observed that as much as 10 to 15% of the carbon within the system was now in the form of the organic compounds. So this particular, dear students, this particular theory, which was proposed by Miller as well as the Urey, it clearly indicates how the carbon-based compounds or the organic compounds were originated in this universe. So it is very simple through this particular demonstration where methane was there, hydrogen and the ammonia and the water, and then through the electrodes lightning type of the sparking was which was arranged. So this is one such experimental experiment which has been universally accepted, and this shows the how the carbon based compounds or the organic compounds were originated in the in this planet. And dear students, as you may be knowing, carbon has 4 valence electrons. Thus, carbon can make a carbon atom can make 4 bonds. And with 4 electrons, 4 valence electrons, carbon can form 4 covalent bond with a varieties of atoms. And this ability makes large complex molecules possible. And in molecules with the multiple carbons, each carbon bonded to the 4 other atoms has a tetrahedral shape, which is showed in this particular slide. That means the geometry it's a tetrahedral geometry. However, when 2 carbon atoms are joined by a double bond, the atoms joined to carbons are in the same plane as that of the carbon. And why is this important? Because the shape of a molecule that actually dictates the reactivity and its function. So students, the shape of any organic molecule or organic compound that actually mainly responsible for the reactivity that will be decided by that decides the reactivity as well as the functioning of that particular organic molecule. So the shape or the geometry of any this carbon based compounds or organic molecule, that is very important. These are you need not memorize any of these things. Just for your information, I am presented different hydrocarbons such as methane, ethane, ethylene. You can see the structural formula and also the geometry which has been proposed for these molecules. And the carbon skeleton, of course, it varies from one molecule to the another molecule. Say, for example, ethane, you can see here, the ethane is there, propane or 1-butene, 2-butene, or it may be with the presence of different branches, such as in the case of the isobutane, you can find there is presence of the branches. And similarly, there may be presence of the rings also, presence of the rings like the cyclohexane, where each carbon atom that is bonded to the 2 hydrogen atoms. And this particular cyclohexane ring, this is completely saturated one, whereas the unsaturated form of the cyclohexane, which is nothing but the benzene molecule, which is showed in the same slide on the extreme right side. And the hydrocarbons, as you know, hydrocarbons are the organic molecules which consisting of only carbon and the hydrogen. So the molecules which are composed of only carbon and the hydrogen are termed as, they are nothing but the hydrocarbons. So in the previous slide, whatever examples I have showed here, you can see the ethane, propane, butane, cyclohexane, benzene, etc. They are basically nothing but the hydrocarbons. And in the structure, you might have observed, they contain only the carbon atom which is bonded to the hydrogen atoms. And there are many organic molecules, such as it may be the fats or lipids, even they also contain the hydrocarbon components. And hydrocarbons can undergo reactions that release a large amount of energy. Even the petrol or the diesel, they are basically the hydrocarbons. So when they undergo the combustion, it releases the heat energy. So this is actually a fatty substance, or we can say the lipid molecule, and this is present on the a part of a human adipose cell, where you can find the presence of the fat droplets, or these fat droplets are nothing but the a fat molecule which is showed in the expanded version. So this is basically, again, it contains the carbon as well as the hydrogen and other atoms also. Students, you might have heard about the term isomers. Okay, let us try to recollect that. As you may be knowing, isomers are nothing but the compounds which contain with the same molecular formula, but structurally they are nothing but the different one. I'll repeat it. They have the same molecular formula. Molecular formula is same, but structure wise they are the different. And in organic chemistry, for such particular phenomenon, we call them as a isomerism, or the compounds which show this isomerism properties are nothing but the isomers. And of course, they have been classified into different categories, such as structural isomers. They have different covalent arrangements of their atoms. Or second one is cis trans isomers. They have the same covalent bond, but they differ in the spatial arrangement. They differ in the spatial arrangements. Similarly, the enantiomers, they are the isomers that are which are actually mirror images of each other and rotate in the rotate light differently. That means the plane of polarized light, one of the enantiomer, it rotates in the right side, and another, its opposite enantiomer, it rotates the plane of polarized light in the opposite direction. So the enantiomers are basically mirror images of each other. Say, for example, here the structural isomer. In this one, you can two different structures I have presented. If you look at the structure is different, but the molecular formula is same here. Or in the second case, in this particular case, cis and the trans type of the isomers. Here you see in the first one, that is cis isomer, the two X could be any atom, or it could be molecule also. So, or any group of atoms. So the two X atoms, they are on the same side. They are on the same side. Whereas in the second figure, you can see in the second diagram, that is nothing but the trans isomer. Here, the two X's, they are lying opposite, or they are at a vicinity. So this is nothing but this particular one is the actually cis and trans type of the isomer. Now you can either your right palm or on the left palm, if you hold it before the mirror, suppose you take the your right palm and just hold it before the mirror, what you'll observe in the mirror, exactly it's a mirror image, right? And both these both these image as well as the your actual palm, these are superimposable, but superimposable and mirror images. Now the same concept, what I'm doing is I'm applying it to the a molecule here. That is, the first molecule is L isomer, and when you hold it this particular L isomer in front of a mirror, there is, you can find in the mirror, you can find it's a mirror image, isn't it? The position of the red colored atom, that will be remaining same, but the other, like HCH3NH2, they move to the opposite sides. That means the CO2H position will be on the same place, but the other atoms, NH2, CH3 and H, they will be gets interchanged. So the first one of the isomer, this is nothing but the L isomer, and another one isomer is the D isomer, that is dextro and levorotatory. Dextrorotatory means which can rotate the plane of polarized light to the right side, whereas the L refers to the levorotatory, which rotates the plane of polarized light to the left side. So this is about the enantiomer. And enantiomers are important even in the pharmaceutical industry. I can say they're very much important because when any drug molecule is synthesized in any pharma industry, these synthesized molecule have they are nothing but like an enantiomer. So one is the D enantiomer and another will be the L enantiomer. So among these D and L enantiomer, both are not biologically active. Only one of the molecule, only one of the isomer will be biologically active. So the two enantiomer of a drug may have a different effects. So one of the enantiomer may show the biological activity or it may be having the curing property, whereas the another its enantiomer that may give them side effect or it may give some toxic effect or it may not be biologically active at all. So usually only one isomer is biologically active and differing the effects of the enantiomers which demonstrate that organisms are sensitive to even subtle variations in the molecules. So these are actually just a couple of drug molecules which have included here. The ibuprofen or the ibuprofen. This is mainly used for pain reliever or as well as an anti-inflammatory drug, right? So here, if you can observe the effective one, the S ibuprofen is effective, whereas the NR ibuprofen, which is the another isomer, that is not at all active, that is ineffective. Similarly, the other molecule that is another drug molecule, which is albuterol, that is widely used for the treatment of the asthma. And here, the R type of the isomer, albuterol, is active, biologically active, whereas the S is not at all having any biological effect or it is ineffective in nature. Let us see what are these functional groups. A few chemical groups are key to the functioning of a molecules. Distinctive properties of the organic molecules, which mainly depend on the carbon skeleton and on the molecular components which are attached to each. That means the properties of a entire molecule is because of the different type of course, to the what are the different other groups which are binded to the carbon skeleton. That is going to decide the actual the functionality or the uh, other biological properties of these molecules. A number of characteristic groups can replace the hydrogens which are attached to the skeletons of the organic molecules. That is what in the pharmaceutical industries, the properties of different molecules which can be varied or the biological actions can be varied or changed or can be increased by removing one hydrogen atom by different other functional groups or other functionalities. And in the organic chemistry, you'll come across with when you are studying the organic chemistry papers, you might have already came across with different type of the functional groups, such as the hydroxyl functional group, which is nothing but the OH, that is alcohol, and then carbonyl, that is CHO, they basically for the aldehydes, and then if it is only CO, then that is nothing but the ketone, and then the carboxyl, that is the COOH group, that is actually for the carboxylic acids. Similarly, the presence of NH2 group, that is the amino functionality, or the phosphate functionality, sulfuryl functionality, that is nothing but the SH. So varieties of these functionalities can be attached or can be bonded to the carbon atom so that the properties of the entire molecule can be changed. Here are the alcoholic group, that is the OH, or the ketone, which is nothing but the C double bond O, or if it is a CHO, then that is nothing but the aldehyde. And similarly, the carboxylic acid, here there is a COOH, you can observe like acetic acid or formic acid or butyric acid, etc. Or in the case of the amines where NH2 is present as an active functional group, say for example the glycine, one of the amino acid where the NH2 group is present. or the SH, which are nothing but the thiols or sulfhydryl. We generally call them as a thiols, or you can also term them as a sulfhydryl groups. Say one of the amino acid by name cysteine, which contains this SH group. Similarly, the presence of the phosphate, which is present in the glycerol phosphate, and then the presence of the methyl group, CH3 group, methylated compound. Say, for example, in the case of the 5-methyl cytidine, where CH3 group is present. So, likewise, varieties of the different functional groups they can be chemically bonded to the carbon atom or carbon containing compounds. And similarly here the ATP, which is one of the important biomolecule, adenosine triphosphate. So this is mainly important for as a source of energy for the cellular process. So one phosphate molecule, in this particular case, you can see one phosphate molecule, and then that is adenosine triphosphate is the primary energy transferring molecule in the cell. And the ATP consists of an organic molecule called adenosine, which is attached to a string of 3 phosphate groups. So that is clear in this particular figure, where adenosine is binded to a string of 3 phosphate containing groups. And the versatility of the carbon that makes possible the great diversity of the organic molecules. Similarly, the variations at the molecular level lies at the foundation of all the biological diversity. So whatever the biological molecules which are exhibiting the different properties, they are because of the variation in the at the molecular level itself. There is a variation in the substitutions or the functionalities. So that makes the variation among the biological molecules. Now, after having a brief information related to the uh, stereochemistry of the organic compounds, organic molecules, and then the functionalities, now let us focus on to the carbohydrates. So students, you may be knowing that all these foodstuffs which have presented in this particular slide, they are basically enriched with the carbohydrates. So let us try to understand what are these carbohydrates, what are their basic function, and how they have been classified, and some other interesting facts which are related to the carbohydrates. So the definition one can give as carbohydrates are essentially they are the hydrates of the carbon. This was actually the older type of the definition, but according to the modern chemistry, the perfect definition for the carbohydrate is they are the polyhydroxy aldehydes or ketones. Carbohydrates are either polyhydroxy aldehydes or polyhydroxy ketones. So in any one of the way, you can define either polyhydroxy aldehydes or polyhydroxy ketones. And they have the following basic composition, that is CH2O N times. And what are the functions of these carbohydrates? Let us try to understand. And the carbohydrates they are the spare proteins, so that the protein can concentrate on building, repairing, and maintaining the body tissues instead of being used up as an energy source. And for fat to be metabolized, that means the metabolism of the fat, the carbohydrates presence is very, very essential. So without the presence of sufficient amount of carbohydrate, fats cannot be metabolized in our body. Please remember this. And if there are not enough carbohydrates, then large amount of fat are used for the energy. I hope you understood this point. Suppose if there is sufficient amount of carbohydrate is not present in the body, then whatever the fatty substances present in the body, which will be utilized for the energy purposes. So the presence of the carbohydrate is very essential. And carbohydrate is necessary for the regulation of nerve tissue, and it is the only source of energy for the brain. So whatever the energy source which is required for the brain, that is actually given by the carbohydrate. Similarly, certain types of carbohydrate, they encourage the growth of healthy bacteria in the intestines, which are very essential for the digestion. So some healthy or some friendly bacteria, we can say, they will be formed in the body, or the growth of such healthy or the friendly bacteria will be generated in the intestine. It is because of the carbohydrates only. Some carbohydrates are high in fiber, which helps to prevent the constipation and lowers the risk of the certain diseases such as cancer, heart disease, as well as the diabetes. Students, now let us focus on the classification of the carbohydrate. Mainly, they have been classified in the four categories, majorly as a four classes. So first one is a monosaccharide. The monosaccharides, these are nothing but the polyhydroxy aldehydes or polyhydroxy ketones, which cannot be decomposed by hydrolysis to give any simpler carbohydrate. That means they are the most simplest type of the carbohydrates. They are the most simplest type of the carbohydrate, which further cannot be cleaved into, which further cannot be converted into any other simpler molecules. So example here, the glucose. Glucose is the example for the monosaccharide, so which cannot be further cleaved into any other molecule. Then the next one is the second category, that is nothing but the oligosaccharide. And the oligosaccharide, in the Greek word, actually the term, this term has, it came from the Greek, where the term oligo, which refers to few or many. That means many saccharides. Saccharides refers to the sugar. Saccharide, the term saccharide is refers to the sugar. So the term oligosaccharide, so the term oligosaccharide, which refers to the many of the saccharide molecules. The next one is the disaccharide, actually, which yields two monosaccharide. So when a disaccharide molecule, when it is subjected to hydrolysis, it gives two molecules. The best example is the sucrose, the table sugar, which is nothing but the chemically it is known as sucrose. When it is subjected to acid hydrolysis, the sucrose molecule, it gives glucose and the fructose molecule. Similarly, the another example is maltose, and when this maltose is present in the germinating cereals, when this maltose is subjected to acid hydrolysis, it gives two glucose molecules. So these are actually typically examples for disaccharides. Students, let us see what are these trisaccharides. Trisaccharide, the term tri refers to the three, and saccharides are nothing but the sugar molecules. So totally, it gives three sugar molecules. That means a large sugar molecule, when it undergoes acid hydrolysis, if it gives three sugar molecules, then that is nothing but the trisaccharide. Similarly, the next classification is, that is the polysaccharide. And the polysaccharides are they are the carbohydrates of high molecular weight, and which yields many of the monosaccharides on hydrolysis. So best example is starch or the cellulose. So if you hydrolyze the starch molecule, it gives innumerable numbers of glucose molecule. Similarly, the cellulose is abundantly present in the green plants. So this cellulose molecule, this is also a polysaccharide, and the polysaccharide cellulose, if it subjected to hydrolysis, this also gives innumerable number of the glucose molecules. Students, you can see here in this particular slide, you can see the structures of the monosaccharide, typically the glucose molecule as well as fructose molecule. And the glucose is an example for the aldose type of the sugar. Aldose sugar means a sugar which is containing aldehyde as a substituent. So you can observe here in this glucose molecule, it has a CHO group. That means it is a aldehyde. So it is a aldose sugar or aldohexose also. And similarly, the second example which you can observe here, that is D-fructose. And the fructose is an example for the ketose. It is also called as ketose sugar because here The second carbon atom you can observe, that is, which is labeled as C2, that is actually nothing but a ketone molecule. It's a C double bond. So it is a D fructose is a keto sugar. And the D versus L type of designation. So this D and L type of designations, these are mainly based upon the configuration about the single asymmetric carbon present in the glyceraldehyde. Asymmetric carbon atom means a carbon atom when it is bonded to four different type of the atoms or group of atoms. So in this example, the glyceraldehyde which is given, you can see the carbon atom is bonded to hydrogen, aldehyde, hydroxyl as well as CH2OH. So here the carbon is it's a one of the asymmetric carbon atom. And in the below mentioned example, the lower representations are Fisher projections. It's a representation for the Fisher projection for the D-glyceraldehyde. Similarly, the Fisher projection for the L-glyceraldehyde. The same Fisher projection, we can extend it to higher members of the carbohydrates also. Say, for example, for the D glucose or the L glucose or the fructose. So, or you name any of the other hexo sugar or hepto sugar, where also the Fisher projection that can be applied and the molecules can be arranged as per the Fisher projection. And the D and L sugars, these are actually the mirror images of each other. Mirror images, and they are they are superimposable. That means they are the enantiomer. They have the same name. Say, for example, D glucose as well as the L glucose. And other stereoisomers, they have the unique names like glucose, mannose, galactose, gallose, and so on. About 8 such stereoisomers are there for glucose. And the number of stereoisomers, how to find out a number of stereoisomers for a given carbohydrate. So please remember, student, remember this formula, 2 to the power n, where n is the number of asymmetric center. Say, for instance, the 6 carbon, that is aldose, which have a 4 asymmetric carbon centers. In such case, 2 to the power 4, that means it becomes 16. So that 6 carbon containing aldose sugar has totally 16 stereoisomers, and of those 16 stereoisomers, 8 are D sugars and remaining 8 are L sugars. Say, for example, here only 2 such D and L glucose has been given. So totally for a 6 carbon containing aldohexo sugar, totally 16 stereoisomers, because as I mentioned, each of these has a 4 asymmetric carbon atom. So when you write it 2 to the power n, that means 2 to the power 4, it becomes 16. So out of that 16, 8 plus 8. I hope this is clear to you. The next one, the 4 and 5 carbon ketoses, which are designated by inserting UL or UL into the name of a corresponding aldose. For example, the D-ribulose. D-ribulose is the ketopentose which is corresponding to the aldopentose D-ribose. Students, these are some of the sugars, that is 6 carbon atom containing sugars, which I have mentioned in the previous slide. The first member is here D-allose, D-allotrose, D-glucose, D-mannose, D-gulose, D-iodose, D-galactose, D-talose. Similarly, it also with the L-allose, L-allotrose, etc. So 8 plus 8, 16 totally. You can see here this molecule, the glyceraldehyde. When you hold this molecule in front of a mirror, you can exactly find its mirror image. So that is the Fisher projection formula. This is the Fisher projection formula for the 6 carbon atom containing molecules, carbohydrates. And now uh, based on the Fisher projection, the next representation for the any such carbohydrate we can do is, that is the Haworth projection. Haworth projection. So in this Haworth projection, it shows the carbon 1 of the Fisher projection is binded with the carbon 5 of the same Fisher projection, and it can be presented in the second structure as per the second structure which is showed in this particular slide. And here look at the position 1 in the Haworth representation. Look at the position 1 where the C1 position 1, 2, 3, 4, 5 and the 6 which represents C1, C2 to C6. At the carbon 1 position, the OH is below the plane. In such case, we can call it as a alpha type of isomer. If the same OH is vice versa, that means OH is on the upper side, then in that case, it is nothing but the beta isomer. So remember, the OH at the first carbon atom, if it is below the plane, then we can call it as a alpha isomer. If it is above the plane, then it is nothing but the beta isomer. Let us see the cyclic structure for the glucose molecule, and this cyclic structure, this has been proposed based on the Haworth representation only. You can see here the glucose which forms an intramolecular, the first structure that is indicating the Fisher projection, and here you can see the C1 of the aldehyde, that means the carbon 1 of the aldehyde group, and C5, that is C5 of the OH, they are reacted together and it forms a 6-membered pyranose ring. So such type of the 6-membered pyranose ring, and that is termed as, it is a pyran. It's a pyran structure. And these representations of the cyclic sugars are called as Haworth representation. I hope this is clear to you. You can see here in the D glucose, the OH is present below the plane. So it is a D. Actually, this is actually alpha isomer. Similarly, on the second one, where the OH is above the plane, this is actually the beta isomer. But the its rotation capacity for the plane polarized light, that is the dextrorotatory only. Either it is a alpha or beta isomer, but both these are D sugars in nature. That means they rotate the plane of polarized light to the right side. Similarly, if you look at the second example, that is fructose molecule, D-fructose molecule, and as you have already seen from the previous slides, fructose is an example for keto sugar, whereas the glucose was example for the aldose sugar. And in case of the, before writing the Haworth presentation, first you have to represent, or you need to understand the Fisher projection. And you can see in the Fisher projection of the D-fructose, the C2 of the keto group, which is making a bonding, or it forms an intramolecular cyclization with the OH of the C6 group. Or there is another possibility also, a 5-member furanose ring by reaction of the C2 with the C5. That means for the fructose molecule, we can present two type of the structure, either the intramolecular cyclization between C2 to C6 or C2 to C5. So in this one, it is showing the C2 to C5 intramolecular cyclization for the D-fructofuranose molecule. Let us see the disaccharide. As you already aware, disaccharides are the molecule when they are subjected to hydrolysis, it gives the two simple sugar molecules. Say for example, the table sugar itself we have taken as an example earlier. It gives the glucose and the fructose molecule. Even the lactose molecule, lactose is commonly present in the, it is also called as a milk sugar. So, or the maltose, which is present in the germinating cereals. So, the maltose is also an example for the disaccharide type of the carbohydrate. The formation of the, let us see the formation of the disaccharides. And these disaccharides are formed when two monosaccharides are joined together and a molecule of water is removed, a process which is known as dehydration. Whenever there is a removal of water molecule, we call such reactions in organic chemistry as a dehydration reaction. Now let us take the example of a milk sugar, nothing but the, in carbohydrate chemistry, we call the milk sugar as a lactose. And this lactose is made up of glucose, one molecule of glucose and one molecule of the galactose. Whereas the sugar from the cane sugar or the our table sugar that is made up of one glucose molecule and one fructose molecule, which I have already mentioned in the earlier slide also. Then the two monosaccharides which are bonded via a dehydration reaction, which is also nothing but condensation reaction, that leads to the loss of a water molecule and it forms a glycosidic bond. That means while forming a disaccharide molecule, with the elimination of a water molecule, whatever the new bond which is formed, that is nothing but the glycosidic bond. Here, in these two examples, the glycosidic bonds are clearer to everyone. You can observe the glucose and the glucose molecule, the C1 and the C4 positions when they are undergoing the cyclization, there is elimination of the water molecule because two OH groups are there, one OH and another hydrogen from the second OH. With the elimination of the water molecule, it forms the glycosidic linkage. That is nothing but the COC bonding, 1,4-alpha glycosidic bond. That is how the maltose is going to be formed in the nature. Let us move on to the next example. Let's move on to the next one. Some of the common disaccharides which we use in our day-to-day life, like the galactose molecule and then fructose. Galactose and fructose, when they are combined together, they form the lactulose. Similarly, the glucose and the glucose molecule, when they are combined together at the alpha 1,4 position, it forms the malose. Similarly, glucose and the glucose molecule, when they are combined together, it forms trehalose. Or just observe here, the linkage is also important. The bond is also important. Alpha 1-to-1 type of the linkage means, in case of the glucose, it gives the trehalose. Similarly, the beta 1,4 linkage for the glucose, which results in the cellobiose. So, it's very important to observe what type of the bonding is present between these monosaccharide units. Otherwise, the monosaccharide units may be the same, but the bondings are different. So, the difference in the bonding that makes the different type of the disaccharide molecule. So some of these are some common disaccharide molecule which I have already mentioned earlier. Now let us focus on to the polysaccharide molecules, and these polysaccharides are actually long carbohydrate molecule of repeated monomer units which are joined together by the glycosidic bonds, and they range in the structure from linear to highly branched structure. Some of the different examples which include storage polysaccharides such as starch and the glycogen. Students, you may be knowing that in case of the plants, the reserve food is in the form of the starch. Say, for example, in the case of it may be from the potato or the rice or the wheat, or you name any of the food, any of the plants where the reserve food in the plant that is the starch. Similarly, in case of human being as well as other mammals also, such type of the reserve food will be there. And that is, but in case of the mammals, it is the reserve food is not termed as a starch, but the animal starch is specifically termed as glycogen. The reserve food in the case of the plant is termed as starch, whereas in the case of the animals, such a type of the reserve food is nothing but the glycogen. And the structural polysaccharides such as cellulose, again it is present in the plants, or the chitin or the chitosan. And this chitosan molecule is actually present in the exoskeletons of crustacean family animals. Say, for example, the either the crabs, the outer coverage of the hard outer coverage of the crab, or maybe the prawns or shrimp, or maybe the lobster, that actually enriched with a polysaccharide which is called as chitosan. And the polysaccharides, they have the general formula CXH2O Y times, where X is usually a large number between 200 and 2500. The general formula can also be represented as C6H10O5 N times. And the polysaccharides, they serve as a stored fuel as structural components of the cell walls and the extracellular matrix. The homosaccharides starch, the homopolysaccharide starch and the glycogen are stored fuels in the case of the plants, animal, and the bacterial cell. They consist of D glucose with the linkage, and all three contain the same some branches also. You can observe here in this particular structure, the starch is made up of amylose and amylopectin, whereas the amylose is a 1,4 alpha 1,4 linkage, whereas in case of the amylopectin, where actually the branches are present. So it's a very large molecule. This is the, you can see the structure of the amylopectin, where the presence of the branches on the main chain. So this is the amylopectin, and amylose is a linear type of the polysaccharide. So starch is mixture of both amylose as well as the amylopectin. Similarly, the glycogen molecule, this glycogen, as I mentioned earlier, it is present in the animals in the form of a reserve food. The reserve food in the animals is nothing but the glycogen. This is basically made up of the glucose with alpha 1,6 linkages. And specifically, there is a presence of the branch at 1 to 6 position. You can observe here in this particular diagram where 1,6 linkage is present in the glycogen molecules. So students, now let us move to the quickly let us move to the next important type of the carbon based molecules or the compounds, and they are nothing but the polymers. As you may be knowing, polymers are nothing but they are the large molecules, or the term polymer that itself is a self explanatory. Poly refers to the many and mer refers to the molecule. That means large number of smaller units or the molecules which combine together, and it forms a single larger molecule called as polymer. So a brief introduction. So during the 1953, Hermann Stedinger, who actually formulated a macromolecule is nothing but a polymer only. Polymer synonymous is macromolecule or large molecular. Macromolecular structure for rubber, and he received the Nobel Prize based on the repeating units of 2-methyl buta 1,3 diene. So this is actually the structure of the natural rubber. 2-methyl 1,3 butadiene is the chemical name of the this natural rubber. And when these large number of smaller molecules, that is monomers, when they undergo the polymerization, it forms a larger molecule that is nothing but the polymer. So the conversion of monomers into the polymer is known as polymerization. This is known as polymerization. And the polymers are compounds which consist of very large molecules which are formed by repeated joining of the many of the small molecules. And the polymers which are naturally occurring, so we can classify them in different categories. One is natural and another one is synthetic one. So as you may be knowing, natural polymers are nothing but which are occurring in the nature, whereas the synthetic type of the molecules, synthetic polymers are those which are man-made or artificially synthesized one. You can see some of the examples such as the proteins. Proteins are actually, they are the basically natural polymers only, because proteins are made up of smaller molecules which are called as amino acids. So large number of the amino acids are undergoing the polymerization reaction, and they form the polymer. Similarly, the polysaccharide. Where cellulose or the starch molecules, you have seen the structure in the earlier slide, which is actually nothing but the polysaccharide. Similarly, the nucleic acids. the nucleic acids such as DNA, or it may be the DNA or the or RNA, that is deoxyribonucleic acid and ribonucleic acid. So these are also considered as naturally occurring or natural polymers. The reason is DNA or this RNA, they are made up of large number of monomers, which are called as the adenine, guanine, cytosine, and the thymine, or the thiamine is replaced by the uracil in the case of the RNA. So AGCT. So that is nothing but the nucleic acids. They are the nucleic acids, polysaccharides, proteins. All they are coming under the natural polymers. Similarly, the rubber. Of course, in the rubber, we can find two types. One is natural and the synthetic. Here, specifically, I have covered the natural type of the rubbers. So, the natural rubbers, which are basically composed of 2-methyl-1,3-butadiene structure. Earlier, the structure has been represented. So, large number of such 2-methyl-1,3-butadiene, it undergoes the polymerization and it forms the larger unit, which is nothing but the rubber or the elastomers. And then synthetic polymers, and the synthetic polymers, which are nothing but they are man-made type of the polymers. They are the man-made polymers, or the other name which we can give is synthetic polymers. And there are hundreds of such synthetic polymers which have been reported or which are commercially available. Say, for example, some of the synthetic polymers such as polyvinyl chloride, or it may be the polyvinyl acetate, or the Teflon, or polysulfone, or polyvinylidene difluoride. So we can give hundreds of such examples under the synthetic polymers. Similarly, the plastics are also, they are also the synthetic materials. In case of the plastics, they have a very high molecular weight. Generally, the molecular weight is in between 70,000 or beyond 70,000. That is in the case of the plastics. And please remember, dear students, you please remember here, all the plastics are nothing but the polymers, but all polymers are not plastics. Even though the terms plastics and the polymers are synonymous, but strictly speaking, the term all the plastics are polymers, but all polymers cannot be plastics. Let us move on to the next part. The example, one more example for the synthetic one is polyethylene. That is one of the most commonly used and the simplest example. Similarly, the polystyrene is the another example. Polyvinyl chloride as a one more example. PVC, commonly we term them as a PVC, or the nylon molecule. Nylon is also synthetic one, or urea methanol, that is also synthetic one. Now let us take few of the naturally occurring polymers as an example. Let us quickly move on to the proteins. So the importance of the proteins are very much required for the as a as a nutritional compounds, and their main function is they catalyze the hydrolysis of the proteins to the polypeptide, and they also catalyze the hydrolysis of starch to the maltose, and they can catalyze the hydrolysis of fats into fatty acids and the glycerol, and also the vital activities such as the respiration, and then cell transport and the immune response, as well as the growth of all these are actually controlled in our body by the protein molecules. Hence, proteins are very much essential for the normal growth. Let us move on to the next one. Let us see some of the protein-rich foods which have in this picture. You can observe the fish or a meat or a milk or the different type of the green peas and other type of the seeds which are highly richer in proteins. And you may be knowing proteins are also very much essential for the building muscles. The proteins are actually large organic molecules with larger molecular masses. And generally, their molecular weights, it may go up to beyond 40 lakhs. For some of the specialized viral protein, the molecular weight may be beyond 40 lakhs. And more typically, several thousands also. It depends from protein to protein. And students, for your kind information, even the even enzymes. Enzymes are also basically nothing but the enzymes are also nothing but the proteins. And the most of the proteins, they also contain the, along with the carbon, hydrogen, and oxygen, most of the proteins also contain nitrogen as well as sulfur, and sometimes the phosphorus may also present. It is not mandatory, but some of the proteins may contain the phosphorus also. And for your kind information, about 20 amino acids which are very important for the human being. For human being, about 20 amino acids are very important. That doesn't mean beyond 20 there are no other amino acids. Actually, about 500 plus amino acids are there, but among those 500 plus amino acids, only 20 amino acids are which are very much important to the human being. And students, for your kind information again, with the different permutations and combinations, if you consider only these 20 amino acids with the different permutations and combination, and then condition is in this permutations and combination, only one amino acid is used one time. In such case, we can get about 2.4 into 10 to the power 8 such a large number of proteins which are possible with just 20 amino acids. I'll repeat again, 20 amino acids with the different permutations and combinations, with the condition that only one amino acid is used only one time. With only these 20 amino acids, with the permutations and combination, it can give about 2.4 into 10 to the power 8. Such a large number. Several millions of proteins are possible with the only these 20 amino acids. Then you can imagine in the nature there are about 500 plus other amino acids are there. With all these permutations and combination, the numbers could be several billions. That's what the protein chemistry is. It's a very interesting topic, or it's under the biochemistry. Okay, let's move on to the next one. That's what I already mentioned, 20 different amino acids. Let's move on to the next slide. And the proteins are long and unbranched polymers of the amino acids. Similarly, the peptides, the amino acids, when they undergo the condensation reaction, they form the peptide linkage. And these peptides are short chains of amino acids. So the sources of proteins, already I have briefed about that. Let us move to the amino acid, which is last part. Maybe another 5 more minutes I'll be taking. Kindly bear with me for another 5 more minutes.
Okay,
So
sir.
in the how many minutes, sir?
Okay, sir. Please.
Okay, yeah, another 4 to 5 minutes I'll be
Okay,
taking
sir.
another, not more than 4 to 5 minutes.
Okay.
So we obtain amino acids from the source for the amino acid is either from the plant or from the animal based food. And our body, which resemble the amino acid building blocks into our body proteins. You can see here in this diagram the dietary proteins, which on digestion they give the amino acid, and then the amino acids, they will, by various enzymatic reactions and the other biochemical reactions in the body, they build the proteins in the our body. And the proteins are basically classified in two categories. One is essential, sorry, the amino acid, in fact, not the proteins. So one is essential amino acid, and the another one is non-essential type of the amino acid. So in the case of the essential amino acid, the meaning is here, they cannot be made in the essential amino acids are not synthesized by our body, but externally they need to be given. So that's the reason they are very much required for the body, but our body is not in a position to synthesize them. So cannot be made in the body. So the term is essential is given to them. And if one is missing, that means if any of the amino acid is missing, then the body proteins are the other body proteins that are broken down to make the new required type of the proteins or the amino acid. Similarly, the other type of the amino acid is non-essential. That means non-essential in the sense our body can synthesize them in a sufficient quantity. So it is separately as an additional supplementary, no need to consume them as an additional supplementary. So essential and the non-essential. And of course, there is another classification of the amino acid is based on their chemical structure as well as what type of the substituents are present, but which we are not highlighting at this particular moment. And these are, I have listed out the essential amino acids as well as the non-essential amino acids. So the examples like histidine, isoleucine, leucine, lysine, methionine, threonine, tryptophan, valine, these are in fact essential type. And the alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, these are all, they are the non-essential type of amino acids. So this is the table which is given. And the protein structure, basically the proteins are made up by the peptide linkages. That means from the when two different amino acids, when they are reacted together, they form C double bond O as well as the NH linkage. That itself is nothing but the CONH linkage or peptide linkage. Similarly, there is a other one is polypeptide, a chain of amino acids which are linked by the peptide bonds, and the proteins are made up of one or more polypeptide chain folded into specific three-dimensional structures. So these are actually the how the amino acids are linked and how they are forming the proteins. This particular slide is showing about the formation of the proteins through the amino acids. And for your kind information, the polypeptides or even the proteins, they can be synthesized artificially also. Also, artificial synthesis is also possible. Let us move on to the one of the important properties of this protein, which is nothing but the denaturation. So denaturation is nothing but change in the three-dimensional structure or three-dimensional shape of the protein because of the various external factors. It may be like a temperature, or it may be the acidic, or it may be the because of the mechanical agitation. One best example I would like to give, the when an egg is cooked or making an omelette. So the omelette immediately it gets solidified, right? The yolk of the egg, when you spread it on a hot pan, immediately it gets solidified. That is nothing but because of the change in the shape of the proteins. This is nothing but because of the increased temperature, the solidification of the or the denaturation of the protein will take place, which results in the solidification of the egg. Or just when you boil an egg also, it gets solidified. The yolk as well as the other part, albumin and the yolk part, it gets solidified. That is also nothing but because of the denaturation. The protein, it loses its structure because of the increased temperature. So that is nothing but the denaturation. So as a result of the denaturation, many of the properties of the proteins are going to be lost. So these are the some of the important aspect which I would like to highlight related to the carbon molecules or carbon chemistry. So that was the last slide. Sir, is there any possibility for the live interaction?
Yes, sir.
Professor, there is possibility if students are willing, we certainly fun.
Yes, sir.
On behalf of KSTA, very thankful to you. This scintillating and informative talk on organic molecules. Covered most of the things from carbohydrate, sugars, then proteins, peptides, etc. I'm sure students would have got a lot of take-home message from your talk.
Thanks, sir. Thank you so much.
They will be benefited by your talk, especially in their PU examinations and also CET and JEE. If there are any questions, students, please ask the question either on chat box, or if you raise your hand, we will unmute you. You can directly ask sir question.
Yes, sir.
At this juncture, I would like to say, sir is the most distinguished chemistry professor in Karnataka. He has been awarded with many renowned prizes and awards, both by Government of Karnataka and other institutes. So if you have doubts, it is the right time to ask. If you don't have any question right now, you may also email us. We'll
Yes,
send
sir.
it to sir for his comments later. Siddhagodra, any questions?
ಕ್ವಶನ್ಸ್ ಚಾಟ್ ಬಾಕ್ಸ್ ಯಾವ್ದು ಹಾಕಿಲ್ಲ ಸರ್ ಸ್ಟೂಡೆಂಟ್ಸ್.
ನೀವು ತಪ್ಪು ಸರಿ ಅಂತ ಏನು ಯೋಚನೆ ಮಾಡಬೇಡಿ. ಏನಾದ್ರು ಡೌಟ್ ಇದ್ರೆ
ಹೌದು,
ಏನ್ ಮನಸ್ಸಿಗೆ ಏನ್ ಹೊಳೆಯುತ್ತೆ ಅದನ್ನ ಬರೀರಿ. ಸರ್ ಡೆಫಿನೆಟ್ ಆಗಿ ನಿಮ್ಮ ಡೌಟ್ ಅನ್ನು ಕ್ಲಿಯರ್ ಮಾಡ್ತಾರೆ. ನೀವು ಹಂಗೆ ಡೌಟ್ ಅನ್ನು ಇಟ್ಕೊಂಡು ಹೋದ್ರೆ
ಹೌದು,
ಮುಂದೆ ನಿಮ್ಗೆ ಎಕ್ಸಾಮ್ ಟೈಮ್ ಅಲ್ಲಿ ಪ್ರಾಬ್ಲಮ್ ಆಗೋ ಸಾಧ್ಯತೆ ಇದೆ. ಇವಾಗ ಆರ್ಗ್ಯಾನಿಕ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅಲ್ಲಿ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್, ಫ್ಯಾಟ್ಸ್ ಅಂಡ್ ಪ್ರೋಟೀನ್ ಬಗ್ಗೆ ಏನೇ ಕ್ವಶನ್ಸ್ ಇದ್ರು ಸರ್ನ ಈ ಸಮಯದಲ್ಲಿ ಕೇಳಿದ್ರೆ ಒಳ್ಳೇದು.
ಸರ್ ಅವ್ರು ಮತ್ತೆ ಬೇರೆ ಏನಾದ್ರು ಸಮಸ್ಯೆ ಇದ್ರು ಕೂಡ ಮುಂದೆ ಯಾವಾಗ್ಲೂ ನನ್ ಕೂಡ ಕಾಂಟ್ಯಾಕ್ಟ್ ಮಾಡ್ಬೋದು. ಇದೇನಿಲ್ಲ, ಇದ್ರ ಟಾಪಿಕ್ ಕೂಡ ಖಂಡಿತ ಮಾಡ್ಬೋದು. ಬಟ್ ಡೆಫಿನೆಟ್ಲಿ ಅಪಾರ್ಟ್ ಫ್ರಮ್ ದಿಸ್ ಬೇರೆ ಕೂಡ ಯಾವ್ದಾದ್ರು ಆರ್ಗ್ಯಾನಿಕ್ ಕೆಮಿಸ್ಟ್ರಿ ಇದ್ರೆ ಖಂಡಿತ ಅವ್ರು ಕಾಂಟ್ಯಾಕ್ಟ್ ಮಾಡ್ಬೋದು. ನನ್ನ ಫೋನ್ ನಂಬರ್ ಮತ್ತೆ ಇದು ಇದ್ರಲ್ಲಿ ಇದೆ, ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಇದೆ, ನನ್ನ ಫಸ್ಟ್ ಸ್ಲೈಡ್ ಅಲ್ಲಿ ಫೋನ್ ನಂಬರ್ ಮತ್ತೆ ಇಮೇಲ್ ID. ಎನಿ ಟೈಮ್ ನೀವು ಕಾಂಟ್ಯಾಕ್ಟ್ ಮಾಡ್ಬೋದು ಸ್ಟೂಡೆಂಟ್ಸ್.
ಮತ್ತೆ ಇನ್ಯಾವ್ದಾದ್ರು ನಾವು ಮುಂದೆ ಕೆಮಿಸ್ಟ್ರಿ ರಿಲೇಟೆಡ್, ಆರ್ಗ್ಯಾನಿಕ್ ಕೆಮಿಸ್ಟ್ರಿ ರಿಲೇಟೆಡ್ ಏನಾದ್ರು ಟಾಪಿಕ್ಸ್ ಇದ್ರೆ ಐ ವಿಲ್ ಅಪ್ರೋಚ್ ಯು ಫಾರ್ ಎ ಟಾಕ್.
ಖಂಡಿತ ಸರ್, ತುಂಬಾ ಥ್ಯಾಂಕ್ಸ್.
On behalf of KSTA, thank you very much. I wish you happy Gauri and Ganesh festival. Please
Thank you, sir.
my greetings to your family.
Thank you. Thank you, sir. ನಿಮ್ ಕೂಡ ಗೌರಿ ಗಣೇಶ ಹಬ್ಬದ ಶುಭಾಶಯಗಳು. ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್.
ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಸಹ ಗೌರಿ ಗಣೇಶ ಹಬ್ಬದ ಶುಭಾಶಯಗಳು. ಮತ್ತೆ ನಾಳೆ ನಾಡಿದ್ದು ಯಾವುದು ಟಾಕ್ಸ್ ಇರಲ್ಲ. ಮಂಗಳವಾರ ಮತ್ತೆ ಮತ್ತೊಂದು ಉಪನ್ಯಾಸದೊಂದಿಗೆ ತಮ್ಮನ್ನ ಭೇಟಿ ಆಗ್ತೀವಿ. ಹಾಗೆ ಈ 2 ದಿವಸದಲ್ಲಿ ನಿಮ್ಗೆ ಆರ್ಗ್ಯಾನಿಕ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಬಗ್ಗೆ ಏನಾದರೂ ಡೌಟ್ಸು ಅಥವಾ ಕ್ವೆಶನ್ಸ್ ಇದ್ರೆ ನೋಟ್ ಮಾಡಿಕೊಂಡು ನಮಗೆ ಇಮೇಲ್ ಮೂಲಕ ಕಳಿಸ್ಬೋದು, ಅಥವಾ ಸರ್ಗೆ ಡೈರೆಕ್ಟ್ ಆಗಿ ಕಳಿಸಿದ್ರು ಅವರು ನಿಮಗೆ ಉತ್ತರ ಕಳಿಸ್ತಾರೆ. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್. ನಿಮ್ಗೆ ಶುಭಾಶಯಗಳು ಮಾತಾಡೋಣ.
ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಥ್ಯಾಂಕ್ ಯು ಫಾರ್ ದಿಸ್ ಆಪರ್ಚುನಿಟಿ.
Thank you, sir. Thank you.
ಥ್ಯಾಂಕ್ ಯು ಸರ್, ಅರುಣ್ ಇಸ್ಲೂರ್ ಸರ್.
ಥ್ಯಾಂಕ್ಸ್ ಉಮೇಶ್ ಅವರೇ. ಥ್ಯಾಂಕ್ ಯು ಸಿದ್ದೇಗೌಡ್ರೆ.
ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಥ್ಯಾಂಕ್ ಯು.
ಆಯ್ತು. ಸೊ ಈ ಸಲ ಕ್ವಶನ್ಸ್ ಏನಾದ್ರು ಕೊಡ್ಬೇಕಾ? ಲಾಸ್ಟ್ ಟೈಮ್
ಸರ್ ಇಲ್ಲ, ಈಗ ಆ ತರದ ರಿಲೇಟೆಡ್ ಏನು ಇದು ಮಾಡ್ತಾ ಇಲ್ಲ ಅವ್ರಿಗೆ ಮಕ್ಕಳು
ಇಲ್ಲ ಅಲ್ವಾ? ಅದೇ ಲಾಸ್ಟ್ ಟೈಮ್ ಅದೇ ಸಮ್ ಕ್ವಶನ್ಸ್ ಅಥವಾ ರೈಟ್ ಅಪ್ ಏನೋ ಇದು ಕೇಳ್ತಾ ಇದ್ರಲ್ಲ ಲಾಸ್ಟ್ ಟೈಮ್.
ಸ್ವಲ್ಪ ಚೇಂಜಸ್ ಮಾಡಿದೀವಿ ಇವಾಗ.
ಓ ಅಚ್ಚಾ.
ಮಕ್ಕಳುಗಳಿಗೆ ನೋಟ್ಸ್ ಬದಲು ಕೂಡ ಇವಾಗ ಕ್ವಶನ್ ಬ್ಯಾಂಕ್ ಟೈಪ್ ಅವ್ರಿಗೆ CET, NEET, JEE ಎಕ್ಸಾಮ್ ಗೆಲ್ಪ್ ಆಗುವಂಗೆ ಸರ್ ಕ್ವಶನ್ ಬ್ಯಾಂಕ್ ಟೈಪ್ PCMB ಎಲ್ಲ ಮಾಡಿಸ್ತಾ ಇದ್ದಾರೆ.
ಹೌದು.
ಎಸ್ ಸರ್. ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು ಸರ್.
ಆಯ್ತು ಸರ್, ಥ್ಯಾಂಕ್ ಯು.
ಹ್ಯಾಪಿ ಗಣೇಶ ಚತುರ್ಥಿ ಸರ್.
ಥ್ಯಾಂಕ್ ಯು. ನಿಮ್ ಕೂಡ.
ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು.
ಯಾ, ಬಾಯ್.